Right. Yes, it's time to kick off our first session for this afternoon. So this is the first of two horizon scanning sessions. And if you look at your program, I should be Dr. Supriya Sharma. Unfortunately, she had to cancel her trip here, so I jumped in to give to do the chairing on her behalf. I hope this is okay with you. And my co-chair is Dr. Bo Lee. And so this first session is on really the idea of horizon scanning, to really see what is at the horizon, what are the new developments that are important and that will impact us over the coming years. And to start the presentation, it's a great pleasure to welcome and introduce to you Dr. Masato Kiyoshi, who is working at the NIHS in Japan. So Dr. Kiyoshi studied pharmaceutical science at Kyushu University, and then he specialized in protein and antibody engineering. So he's a protein scientist, and currently he's working at NIHS on the molecular characteristics of therapeutic antibodies, looking at structure, function, and therapeutic potency, as well as stability. So over to you. Thank you. In fact, yeah, sorry, I've forgotten the title of your presentation is Structural Analysis of Therapeutic Antibodies Using Cryo-EM.
Firstly, I just want to say thank you to everyone here. Thank you for organizing this wonderful conference, and thank you for having me here. I'm Masato Kiyoshi. I'm a scientist of National Institute of Health Sciences in Japan. Today I'm going to talk about structural analysis of therapeutic antibodies using cryo-EM. So recently, the number of biologicals has increased rapidly. So here I'm showing the number of biologicals approved in Japan. Then biologicals include the hormones, cytokines, enzymes, blood coagulation factors, toxins. And here I emphasize that green shows the number of antibodies, and dark blue is the antibody drug conjugates, and then brown is FC conjugates. Then total number of antibody drugs is approaching about hundreds. It could be more than 100. So here I'm showing the structure of therapeutic antibody drugs. The IgG antibody has two FAB domains that binds to the antigens and one FC domain. The FC domain is really important for FC effector functions. And FC has N-glycosylation site. You can see that green that one is N-glycan. And hinge region is also important for function, and that connect to the two FAB domain and one FC domain. IgG has two heavy chains shown in blue and two L chain, light chain shown in gray. IgG has Y shape. So here I'm showing the three major mechanisms of antibody-based cancer therapy. The one is CDC, complement-dependent cellular cytotoxicity. And complement can destroy the cancer cells. And second one is ADCC. ADCC, this is the most important antibody-dependent cellular cytotoxicity. And natural killer cells kill the cancer cells using granzyme and perforin. And third one is the ADCP, antibody-dependent cellular phagocytosis. Macrophages can kill directly the cancer cells. So here I'm showing the metrics for therapeutic antibody approved in USA and EU. On the above, antibody formats. So we can see that not only conventional IgGs, various antibody formats are being developed so far, such as ADC, antibody drug conjugates, and fragment antibody, bispecific antibody, IgG mixture, fusion protein, and others. And in below, disease areas. In addition to oncology and inflammatory diseases, not only that, various diseases are targeted for antibody therapy development, such as infectious diseases, COVID, or Zika virus, or Ebola virus, hematology, neurology, and metabolism, and ophthalmology, and musculoskeletal, or transplantation. So now it's expanding. And here I emphasize that most of new therapeutic antibodies are engineered, such as mutations, domain swapping, and conjugates. Here I'm showing the therapeutic antibodies currently in clinical trials. In the left side, non-cancer indications, and only 33% of antibodies are not engineered. Others are engineered, such as afucosylated, that is sort of, and glycan engineering, and FC science, half-life extension, hinge stabilized, that is IgG4, and some others. Then on the right side, cancer indications, only just 18% of antibodies are not engineered. Here I'm showing the examples of engineered next generation antibodies. Then various molecular designs are developed by protein engineering, such as bispecific antibodies and dual variable domain Ig, DVD Ig, we can see somewhere, wherever, but Ig single chain fluid. So here this is the aim of this study. Unlike the conventional antibodies, like left side, so for which platform technologies has been well established. Compared to that, in the case of next generation antibody drugs on the right side, it is required to establish efficient quality assessment methods. So we had a problem. We don't know how to assess this quality of next generation antibody drugs. It's getting more and more complicated. Especially the structure of therapeutic antibody drugs are not evaluated in detail in CTD, Common Technical Document. So we have a problem. There's a strong need for development of structural analysis methods that incorporate advanced new technologies to assess purity, structural heterogeneity, and stability, thermal stability. Historically, in general, we have three major methods for structural analysis of proteins. We used to use X-ray crystallography, and the other one is cryo-electron microscopy. The third one is NMR. We used to use X-ray crystallography, but in X-ray crystallography, we have to obtain proteins, and second, we have to purify them. Then we have to crystallize them. Then we have to perform X-ray diffraction experiment. And finally, we can get crystal structure. We can determine the crystal structure of protein. Compared to that, in cryo-EM, we use grid. Then proteins are there. Then we irradiate electron. Then we take pictures of proteins. Then we can get structure of protein by projection. So you have to take pictures of protein from various angles. In cryo-EM, we don't need to obtain crystals, and also we don't need to go to synchrotron radiation facilities. So it's easier. So in this study, as a first step, we try to perform structural analysis using three model antibodies. That is, we selected rituximab and mogamulizumab and trastuzumab, three conventional basic antibodies. This is cryo 200. So we have two major methods of electron microscopy. One is the vitreous ice embedding method. In this method, the protein, we can see the yellow one is protein and in buffer. then we use grid, then we irradiate electron, then we can see that proteins in dark, the background is white in this method. The other one is negative staining. We use uranyl acetate so electron cannot go through metal. So in this method, the background is white, dark, then high contrast. But in this method, this is not inaccurate solution. So first, we tried vitreous ice embedding. Then I said it's easier, but it's not easy actually, because the obtaining a high quality grades is a key step. Because the cryogrid preparation is a trial and error process that is affected by diverse factors such as grid material, incubation time, and blotting. So figure B is the ideal, good condition. This is super fantastic. Sometimes we fail because in figure C, preferred orientation. So as I said, we have to take pictures from various angles, So this is preferred orientation. But in figure D, denaturation, proteins already broken. And figure E, disassembly, some domains are missing or something like that. And figure E, adsorption on film. And G, aggregation. These are bad, sucks. Then, yeah, this is important. So somehow I tried to observe protein using vitreous ice embedding method. Then we observed antibody molecule successfully, but without aggregation. But the contrast was too low. On the left side, we can see that dark particle. This is very too small, but this is the antibody molecules. But contrast was too low, so we quit. So now we moved to negative staining. This is the high contrast. So this is the flow chart of negative staining method. This first we have to take pictures, a lot of pictures. Then picking up particles manually. It's about 50 to 100 particles. Then we use that images. We have to obtain an image for auto picking by superposing them. Then you can see that dark white particles. This is the model. Using this model, you have to auto picking from 100 images. This is just a one image. We have to use a lot of data. Then they pick up all the particles, and we have to build a two-dimensional structure. This is by superposing. Sometimes we repeat this procedure. In this method, we optimize the parameters for picking up particles. But the background noise was reduced. On the left side, this is the previous image, but too much particles we obtained. But the parameter is the picking up threshold was 0.01. This is too much, too low. So we changed the value to 1. Then distance was the distance between two particles, 160. We changed that value to 180. So we could noise, background noise was reduced. So this is our result, and in the rituximab, we got 40 models of structural variation, and mogamulizumab, and 11 models we got. Then trastuzumab, 26 models we obtained, but you can see that trastuzumab, its images are obscured, not clear, not sharp, so resolution was relatively low. Then these results suggest that structural variations could be discussed by comparing these images. Could be. So we found that antibody molecules are highly flexible in solution. Then we successfully observed FAB and FC domain clearly. You can see that two FAB domains and one FC domain. And in the center, we can see dark hole. But these structure models have diverse angles between FAB and FC, diverse angles. And also we try to construct 3D structure by these images. You can see the two FAB domains and one FC domain, and hinge regions are connecting them. But this is the resolution is too low. Resolution is 34 angstrom. So now in summary, we obtained the images of antibody by negative staining. Then we successfully observed antibody molecules. Antibody molecules are highly flexible. The structural variation could be discussed by comparison of these images. Then we have to increase the resolution or try the BTRS ice embedding method. Of course, I have to try to use next generation antibody, complicated antibody. Thank you so much.
Thanks for Dr. Masato Kiyoshi. Any questions, please?
Thank you very much. Yeah, I would have a question. Is any scope in using unnatural amino acids to get incorporated into antibodies to change their properties, also their stability?
Yeah, I completely don't know how, but if the non-natural amino acid is incorporated in structure effect or stability, or I don't know about it. So that is an important question. So you have to take pictures of engineer antibody. Thank you so much.
Any more questions?
Thank you. And I must apologize, I was finishing something, so I did not catch your whole presentation. But I suppose that the type of resolution depends upon the question that you're asking. So you already can see the motifs of the main domains of the antibody. I mean, you clearly will never be able to attain the same level of spatial resolution that you can do with X-ray crystallography. So what are the questions that the resolution that you currently have do not allow you to address?
Intrinsically, using negative staining method, the resolution is maximum 10 or 18 or 15 or like that. So we have to, certainly we have to try to use the visual science embedding. Or how about if we accumulate images, a lot of images, so how about the resolution is what's going on about. So I don't know how much we can increase the resolution using.
I don't think I was clear. No, it's not so much about increasing the resolution. Is that, for example, I find that what you got is already really, really nice. But are there is there specific information that you are missing from what you can do with the current cryo-EM techniques that you've showed us?
What is missing? So I think we can use these images to correlation with stability or aggregation tendency. So we just want to know how much this technology can be used for structural analysis, then correlation between other characteristics of antibodies.
Okay, thank you. Thanks.
Okay. Thank you for Dr. Masato.
Thank you.
Thank you. So the next speaker is Claudius Ginsinger. He working for the European Commission's Joint Research Center. He worked on biomedical testing methods. Since 2022, he is a member of the leadership team of the GRC's Project portfolio on innovation in life and health science. Claudius has a neuroscience degree and got his PhD in Max Planck Institute. So, Claudius, over to you.
Okay, thank you very much for this kind introduction. Thank you, everybody, for being here, and thank you for giving me the opportunity to talk to the organizers. I'm stepping back. So we had super interesting talks, very technical talks in the AI session. I'm stepping back a little bit. I'm not sure also I'm doing horizon scanning really, but anyway, let's see. I would like to start by asking you kindly to just close your eyes for a second and imagine the famous sculpture, The Thinker, Le Penseur by Rodin. You all can do that, right? Of course you can do that, because we're humans, we can imagine. If you ask an artificial intelligence system, of course it can't do that. I don't think everybody would think it can do that, not yet, although that was the major promise at some point. So I would like to take that little example as sort of a categorization I do with regard to the societal perception of AI. One is the very deep fear that I was just talking about. Can machines think? Alan Turing's famous article, also that promise that is still lurking around, artificial intelligence. And then with the rational fears, of course, and the unbridled enthusiasm. And I think we have to navigate between the rational fears, what it will do with our societies, with the decision-making, and the unbridled enthusiasm. And I'll talk a little bit about this. So, very brief, I think it's also nice to remind ourselves of the developments that were really quick, actually. If you think about the first stage, in a way, for AI could be called the cybernetics age, which looked for a natural science of perception of thought with the McCulloch-Pitts neuron, or the famous paper by McCulloch-Pitts. One a neuroscientist, by the way, the other one a philosopher, a logician. So already interdisciplinary at the time. That also kicked off then the John von Neumann computer that we're still actually using in design. Nothing has changed. The second phase, you could say, is cognitivism with cognition as a function of the brain. It was really basically the first foundation of the cybernetics age, and saying, okay, maybe actually the brain is like a computer, and actually we're just manipulating symbols in our head. To some extent, you could say that the symbolic AI maybe is related sort of to this second phase. And the third, of course, is the one that I'm very happy about as a neuroscientist, and it's basically the connectionism. That means the brain self-organizing through Hebbian learning rules and creating feature maps in an activity-dependent way. And of course, groundworking work was done in the visual cortex. And actually, the visual cortex is still that example for convolutional neural networks. We shouldn't forget that. So the work by Thorsten Wiesel and David Hubel for ocular dominance selectivity and orientation selectivity. The perceptron in 1957 by Frank Rosenblatt is also an interesting thing. It was basically a neural network with one layer. We shouldn't forget that, but it didn't work very well because it was just too simple. It was basically like a retina. But we are there. We have now neural networks, and they are getting faster. Thomas has very nicely outlined the power increase that we have. And of course, we have continuing innovations. Huge multimodal data sets are coming. Data sciences progress, and we have new computing techniques like the quantum computers, etc. So it's really, I'm talking about here about AI according to the quote of Marvin Minsky, really from the point of view, what is it doing? And it's in a cross-cutting area of regulatory science, really. It has to be, because it's so horizontal, of course. So one word about intelligibility, explainability. I have some quotations here. There's a false hope of explainability, or don't give up on it, or there's not even a consensus on what explainability is. I would like actually to start a little bit lower. Maybe it's not so much the explainability at the moment we should worry, but maybe we should worry more about the way things are reported, the clarity, the transparency of what data have been used, where do they come from, how they are described, how they have been procured and processed, etc. So we did a study on COVID and AI systems, and we found really a very inconsistent vocabulary study design. And there is also a very nice paper by Nagendran on a systematic review of AI versus clinicians with a very poor trial design that actually favored a bit AI studies. The background, I'm skipping a little bit, our work in the JRC is very much geared towards innovation in life and health sciences. We have different projects there, and we are working in our small team on artificial intelligence mainly. So when I talk about AI in healthcare and medicine, I mean principally a lot. Of course, there's the healthcare with a diagnosis, there's the clinical care, there is also the health system management in principle, there's a health research that's super important, integrating lots of information potentially, the electronic health records, European health data space coming up, and of course there's also in principle public health and surveillance. This system is based on the WHO paper on AI in health. It's very useful. We are focusing more on the first topic on healthcare, actually. And I would just like to make the point that maybe we're a bit in a limbo situation at the moment, because we have there's actually one mistake. Maybe I can go back. No. Okay, there is a mistake in this slide. Anyway, the mistake is just that I'm missing one slide, but we have a very complex system of legislative, don't know why that's missing, legislative acts. We have the AI Act, we have the Cyber Resilience Act, we have the NIS directive in Europe, we have also, of course, the GDPR, we have the European Health Data Space, we have all sorts of, a whole ecosystem, and it's very complicated. The AI Act is very horizontal in a way, because it points then also to the MDR, the IVDR, the Personal Protection Regulation, and other directives. There is a draft commission mandate for the to send Senelec for standardization, but of course the standards really still need to come. There are in the mill, and there is still an ongoing discussion actually about the relationship between horizontal standardization and vertical standards or sector-specific guidance. So there is also something which is emerging only. And the third element that's emerging is consensus-based guidelines. There are a lot of them actually for validation, so to speak, or clinical assessment. We have the dark blue ones out, others coming up, and some other tools that I would invite you to look at. That's very, very interesting and in principle helpful, but they're a bit more sort of crowded towards that step of clinical assessment, which is super important, but it's not always entirely sufficient for developers and also for downstream users. So what our point really is that we want to basically cover this life cycle of and turn it into an evidence pathway and look at different communities, developers, users, health system operators, HTA assessors also. Look at what's happening with the standards and also see that for the good science we have a lot of distributed information. People are not aware of some of the very useful tools that are also published simply in scientific papers. It's difficult to find. So what we want to do is to do something very simple, actually. We want to provide a pragmatic toolbox for describing AI systems, communicating key elements, and helping people to evaluate not only the risks, also the benefits, because that's very important down the line when you're, as a developer, coming and you want to have health reimbursement schemes from HTA processes. You have to make a convincing case that it's not just, it can be done, that's nice, but is it better? Is there a value for the system? So what we're doing very simply is we are basing our work on case studies to understand a bit better what's going on in the upstream element of the research development space, and then also in the deployment space. The FDA has a very nice list of medical devices that are AI enabled, and we are analyzing this. We're developing an ontology, terms, relationships, contents, vocabulary for communication, and we are looking into or working on a classification description matrix, because there is one from the OECD, but it's super horizontal. It's basically for general AI applications. So there is one needed for healthcare, and we're trying to work out that one. And the third element in this toolbox is then a pragmatic approach for exploring potential benefits and risks. The remaining time, I'll just getting you a quick overview. So we'll have built a JRC database also for the ontology and for the case studies that then inform the work that we do on the classification, of course, an ontology, because it has to be grounded in real world existing systems. What we did was something very simple. We did an interrogation of papers on AI in healthcare regarding some key descriptors about the system, about the field of application, the users, data, where do they come from, the model output, and also a bit the use of terminology. Very simple study. And What we also then did, we of course looked what are these papers about. So we had this categorization of aboutness, where we say development or use and assessment, which is surprisingly little actually. I was surprised that we wouldn't find more, for instance, of clinical assessment of some of the tools. And what you can see here in this matrix, it's not really clear, of course, but the main information is actually that we found that there's very scarce information on users' handling of data, privacy, and performance monitoring metrics, especially for the post-deployment stage. That people think ahead, actually, what are the metrics that I would need for that type of system. The other element, I'm not talking much about the ontology. I'm just showing you the high-level graph that we have that is basically work in progress, but it should help, as I said, to facilitate identity analysis, common vocabulary, knowledge sharing, and transition from design to real-world use. That's a focus in the green boxes, which is difficult to see. Apologies. The classification description matrix is based on various sources, our own reflections, but then also work we have done in the past on the IMDF health impacts, where we had a leading hand in developing the adverse event terminology, which we would like to propose actually that it's being used also for that. It's basically could be part of an ontology for that. We use the OECD framework, trustworthy AI principles, and also the paper from the European Parliament, from the STOA group, which is very, very useful. And many other publications, of course, in scientific papers. With 5 modules: AI system, health application dimension, operators, users, stakeholders, human agency, and then very important, the input data, the training data, the knowledge bases, where do the data come from, how they're curated, etc. And last, the algorithm, the model. And what is very important also, and I think a bit undervalued, so to speak, is that people maybe don't spend enough time to explaining what is the relevance actually of the system. Why, how is it scientifically coherent to the data that we have? Why is that a model that should be used? Why is that even an algorithm maybe that should be used? Why not another one? I think one of the session chairs made the nice remark that you don't always have to go probably to a convolutional neural network. You can also use actually more classical statistical approaches, and they're also fine. So the last element then is this risk-benefit evaluation pathway, where what we want to do with that, we want to provide charge questions for developers, but also for other user communities, to basically trigger, for the people to reflect on whether they have fulfilled specific requirements that are necessary in a forward-looking way. We provide explanations, references, and proposals on how to address specific topics, and also examples for actions. And it's very simply structured with 4 steps: gains, benefits, adversity, and risk. These are substructured then in the yellow fields that you can see there. Then benefit risk profile iteration that people should do over the process. And then very important from our point of view, the monitoring, surveillance, and mitigation and responsiveness, which is one of the principles also from the WHO. So this is summarizing a bit the idea behind it. So what we want to do is to provide this toolbox for the different communities, that they can talk to each other, but also that developers actually are thinking ahead. Because often people come and say, yeah, I've validated my model, and that's fine, but that's not enough. Of course, if you really want to bring it on the market, you need to validate it actually in a much more extensive way. You don't need to do user research. You need to do human factor validation, basically. You need the clinical validation, of course. You have to think about the metrics, really, especially also for explainability, where there are issues. And we cannot explain everything. And I would also think that we are maybe emphasizing this point of explainability. We're not so strict with human actors when it comes to explainability, but we're very hard with AI. I think it's more important maybe to ask AI developers to be clear of how the system was developed, and especially where the data comes from. We're in a different world now with the neural networks in any case. That don't work. You cannot have the explainability probably that you could have for statistical model. So you can then apply this over the pathway, this pathway. And what I would like to close with really is that for the explainability, I'm not arguing that there shouldn't be new methods looking at explainability, but I think maybe it's actually more important at the moment that people are thinking ahead, that they're doing user research, that they're doing real-world setting considerations, that they think about the clinical trials early enough and use the tools that are there, that they think about post-deployment monitoring, metrics, and the responsiveness also. And that will build trust. And I think that's probably the first step we should move into. With that, I would like to acknowledge the people, Hubert, Alex, Persa, Uwe, and Vittoria, Hubert, Sander, and Thorsten Prinz and Hans Wenner that are working at VDE. And the last thing I wanted to show you is that we have a paper out there that's nice. It was a PSRS workshop on data quality for AI. There's also a section on healthcare medicine, which we wrote together with the panel members. And it's very high level, but I think it's a good start actually in that field. Thank you.
Thank you very much, Claudius, for a very inspiring talk on AI and healthcare and neural networks. Do we have any questions? We have a bit of time for questions.
Yeah, very nice presentation. You know, you brought up a really good point about the, explainability and trust building and that sort of thing. And it harkens back to doing good laboratory practice studies and experimental design. And in order for your work to be accepted, you really can't change your experimental design during the study. Okay, I mean, you say what you're going to do, then you got to do it, and then you got to publish it or at least get it reviewed. And so that raises the question that you did, is that how do we use that same sort of very regimented fashion when you're talking about using possible programs that evolve over time and improve and that sort of thing. So we want the improvement, we want to be able to capture all the nuances and have them think much better than we can, but that means they're probably changing along the way. So how are we sort of going to deal with this philosophical issue of rules as they have been, and perhaps we need new rules, but how are we going to do that?
Yeah, it's an excellent question. And so I mean, maybe I alluded to it a little bit during the presentation. What we're trying to do here is to have clarity and guidance in a forward-looking way for the developer, but also to connect the communities for people, for instance, that procure systems at the end of the day or that have to do the health technology assessment. But that's basically the pre-deployment framing phase, I would call that, where you frame and you provide the evidence along the pathway. But you're absolutely right, you're not done. Because once you go into post-deployment, it's like for medical devices as well, but for algorithms it will be much more like this. You have to constantly monitor, and you also have to fine-tune, because they're probably otherwise deteriorating. There are also reasons in technical progress, for instance with image quality, that you have to really update your system basically to deal with the better resolution images. Just a trivial example, but it's one of those. So what we believe in a nutshell is that the post-market activities and monitoring, that must be much closer, must be much tighter in a way by the developer also, and we need better frameworks actually for tackling this. That's also why we're trying to connect it to the IMDF adverse event terminology for high impact classification, that there is a sort of an ontology of medical effect terms that people could use if there was any effect, or what sort of effect were there. That could then help describing and exchanging information across regulatory authorities. But the post-market, the post-deployment elements, in my opinion, becoming much, much more important even than for products that are somehow frozen, like a hard valve. They're okay if the production is clean and proper, yeah. But you don't need to. You need to monitor that they're doing well, of course, but you don't need to. They're not evolving in a way anymore in the post-market space. Good question.
Thank you. Any more questions?
Do you have time for questions? Okay, so, and we discussed a little bit about the explainability, and along this line, there's many different metrics that has been discussed about how to judge the trustworthy of the AI. But sometimes we sort of did not really emphasize too much on the context of the use. And for example, in the regulatory application, maybe sometimes false negative does not have too much concern because, eventually a human going to go through the whole thing, and even there is some sort of the mistakes also bring it in, it'll be fine. And other application may be a little bit more stringent in terms of the explainability or reproducibility, so on and so forth. So I just thought maybe, do you have any thoughts on along this line?
Well, I think on the explainability, again, if you if you ask a radiologist or if you ask a medic how he comes to his decisions, you will find that there is a lot of also hand waving at the end of the day, and there's a lot of intuition, which is not wrong, because I mean there's a very capable computer after all, and it's not a computer, by the way. So, okay, but I think the issue really is that if you're working with these neural networks, you will hit a wall probably of explainability. Again, I mean, as a neuroscientist, we had people, when I did my PhD, with people in our group that were doing modeling neural networks of the cortical, one column, basically, to understand whether our assumptions of the cortical column, how the emergence of feature detectors come about, whether they're correct. And we have now an explosion of neural networks in another way. But in neuroscience, we still don't understand at all, actually, how these columns operate really and how they come into being. There's an interesting project by Henry Markram at the EPFL in Lausanne that you certainly know about. Maybe there will be more input from neuroscience, I could imagine, over time, also again in computer science and vice versa on that issue. There are some people who are trying Fourier transformation now to see whether they can explore a neural network basically in how it was set up and developed over time. But I think we shouldn't throw the baby out with the bathwater. That would be my plea, because I think we're using also pharmaceutical substances. We don't fully understand some of them, how they actually work. But we trust them because we know they work, and we have monitored them very closely and found that there were no adverse effects, or we can manage the adverse effects. And I think we have to be a bit careful that we really don't use too different a scale for AI than for human actor. It's very interesting. We always compare, and we should be very careful. I think that's a point also Thomas made. I agree with that. That we don't try to sort of ask for the same mistakes to be made by the AI than the humans. So that would be my answer.
Okay, thank you very much for your contribution. And now it's my pleasure to welcome to the next speaker to the stage, Dr. Jean-Loup Dorn. So Jean-Loup Dorn, he works in EFSA. He's been with us since 2006. His background is very much pharmacology, pharmacokinetics, drug metabolism, and he worked for a considerable number of years in Southampton, and he brought all his expertise to EFSA. And in EFSA, he's been very instrumental in a number of projects, a number of important projects here, such as EFSA's Open Food Tox database, TK Plate, harmonized method for mixture risk assessment, and I'm sure you're going to share some of that information with us. Over to you, Jean-Loup.
Well, thank you, George, for a very nice introduction, and thank you for inviting me and giving me the opportunity to finally take you through the TK Plate platform. It took about a decade to develop this tool. I'm sure it's completely imperfect, but maybe I wanted to start with there is a little joke behind TK Plate, because when you go from exposure to effect, and you're eating, you need a plate to eat. So this is the bridge between the external dose and the effect. So I'm going to go through some generalities first and you've heard a lot about NAMs in details. This is quite high level. And then I'm going to talk about the different models that we have around in the world and how we've implemented them into the TK platform, and just a very few future perspectives on the platform. So you all know about NAMs, but one of the things that we got interested in is also the historical development in mode of action, adverse outcome pathways, and the argument that adverse outcome pathways do not contain kinetic data because kinetic data are chemical specific. Well, I actually disagree with that, because metabolism is like receptors, and there is now the reunion of the kinetics within the adverse outcome pathway framework, and that's the Justin Teagarden's paper and Cecilia Tan's paper on aggregated exposure pathways. So really, this is to help us to go from external dose to understanding the adverse outcome with all these different steps. We have published a guidance document on scientific criteria for grouping chemicals when we assess mixtures, and of course the highest level, or the most refined level we could do that, is to take these processes into account. So I don't think I need to explain you what NAMs are, but okay, in vitro, in chemico, in silico. and I think I will go to the next slide. Something that also I think Maurice mentioned during his presentation. Of course, the chemical strategy for sustainability does mention alternative testing methods, and I think at EFSA we're quite active into this. There's also this one substance, one assessment position paper. Recently, EFSA published, well, in partnership with quite a number of academic colleagues and colleagues from public agencies, a roadmap on NAMs. And as you can see, 6 areas were prioritized, including human susceptibility, also mentioned by Nicole this morning, human population viability, toxicokinetic assessment, cell models, including organ-on-a-chips, development of additional AOPs, exposome data. And of course, the data integration is really quite important, as highlighted again by Nicole this morning. So going into those models, this is a very basic slide, but it's always useful to go back to the really basic stuff. When we think about what we have in risk assessment, sometimes we just have an exposure and a toxic response, an OAL in the rat, and we have an exposure estimate. In food, we have the occurrence data and the consumption data. We combine it, that's our exposure, and then the OAL in the rat. Well, ideally, we want to start taking into account all these biological processes, all the way down to the so-called biologically based dose-response model. And in principle, is to connect exposure, internal dose, and toxicity in a quantitative continuum. So, first thing, we wanted to develop generic models that would have a broad application in the context of EFSA's remit. So the first thing we do is to collect quantitative physiological data for the species of interest. Let's say I'm interested in the rat, or the rabbit, and so on. Chemical-specific parameters, of course, thinking about case studies already. We then integrate these into an algorithm, which is basically a set of differential equations, and then it becomes what we call a physiologically based kinetic model. Something very important to look at for these models is what are the variables that are influencing the impact, impacting on the model, so sensitivity, variability, and uncertainty analysis. To mention the recent guidance document from the OECD on the use of PBPK model in regulatory settings, one of the key issues, of course, is to be able to compare available experimental data with your predicted values. Finally, once we've developed those models, we'd like to put them into an open source platform for users where they're integrated into a user-friendly platform, and of course try to predict kinetic and dynamic properties, and hopefully this might help us to accelerate the risk assessment process without doing testing. So, of course, one of the applications, and again, I think that was discussed also with Nicole with the ICE, we have kinetic variability. So the origin of the data on what we so-called pathway-related variability is coming from the pharmaceutical database, because we do have a lot of pharmacokinetic data. So we may run what we call the quantitative in vitro, in vivo extrapolation model, in vitro metabolism data combined with human variability, run through the PBPK, applied to a specific chemical, and here we can generate a number of outputs. For example, population-specific simulations, let's say comparing adults and neonates, integration of kinetic parameters in the risk assessment process, so in the hazard characterization, internal dose estimations for target organs. I'm interested in concentration inside the liver for this compound, in vitro, in vivo extrapolation for effects, in vivo dose response modeling based on target organ concentration. So this was also stimulated by the new regulation for pesticides, 283284 in 2013, which now requires comparing in vitro metabolism between test species and humans. Of course, if you have a disproportionate metabolite produced in either species, it may have a consequence on the toxicological assessment, so we need to know that. We still have the in vivo study in the rat, of course. But then if we see differences, then we will have to take this into account in our risk assessment. And here is a scientific opinion published a couple of years ago, and an external scientific report associated with a number of publications where we try to combine these these things together for case studies including pesticides and contaminants. So the bottom line here is let's try to take into account variability in metabolism within the PBPK models. So, for example, if we are interested in using historical data and compare the rat with humans, we have a number of steps from which we could derive all the way a human internal, we call it reference point, but of course, it's also called the point of departure. This is the OECD guidance, and if you're interested in the topic, there are a lot of annexes with, I think, something like over 30 case studies. We've participated to 3 case studies in humans, fish, and farm animals. There's one on read across. There's a lot of material there. So these are the generic PBPK models, so examples for farm animals. Sensitivity analysis at the bottom of the slide, and on the right-hand side, comparing predicted concentration with observed concentrations from the literature. Toxicodynamic models, benchmark those modeling, used a lot. We had a guidance document at EFSA published in 2017, which used frequentist methods. This year, 2022, the guidance document is proposing to use a Bayesian approach. Talking about toxicokinetic, toxicodynamic models used a lot in environmental risk assessment. We have, for example, the GUTS models, the DEB models, DEB-TOX models, and this is associated again with a scientific opinion on TKTD model effect models for aquatic organisms. Now, we explored the use of what we called DEB models, dynamic energy budget models. So basically, you have information on the life cycle of an organism, how many eggs it lays, and so on, up to the adult stage. These are fed into a dynamic energy budget models, and the inputs that are required to run this model are toxicity parameters such as no effect concentration, elimination rate, so some feel of kinetics, and killing rate for the survival only. From these models, we can look at effects on individuals, population as well. So this is the TK plate platform, in which we've introduced these kind of models. 2020 was the prototype, and here is the workflow. You have an input module, you have a number of models available, and then you have an automated report that is generated together with graphs and data. So we just try to visualize it. So very important to think about the relevance of this for EFSA and for other agencies and the scientific community in general. Of course, all units dealing with chemicals at EFSA may be able to use it. I do hope so. Supports the use of NAMs in EFSA's business as usual without testing. We can generate TK or TD profiles in minutes. Identify data gaps, of course, because we do have a lot of data gaps. And just some examples: pesticides, food and feed additives, contaminants, novel foods. The PBPK and the QIVV are particularly relevant to pesticides. For example, we're thinking of in vitro batteries for DNT, developmental neurotox. If we understand the consequence of metabolism, let's say a bioactivation to a toxic metabolite, integrating these aspects would be quite critical for the quantitative assessment. When we think about inputs, where can we get the data? Data from regulatory dossiers, kinetic data, dose response for regulated products, for example, literature, previous assessments, and of course public databases such as ECHA's REACH database, EFSA's Open Food Tox, and of course the US EPA Comtox Chemicals Dashboard. So, excuse me, we have an info page which basically gives you an overview of the platform. It should be published in the next few weeks with an editorial, an external scientific report basically explaining how it's been developed, a technical report which is a user manual, module by module, dedicated publications with case studies for each module, and we will run training courses for EFSA staff and experts. So, this is the input module. So I want to run the pig model, the swine model. I'll choose the model, and I want to run it for chlorpyrifos. I choose my chemical, I choose my exposure and time scale, 24 hours or 1 dose, 5 milligram per kilogram. And I then generate my input parameters. And from that, I can run forward dosimetry, so from external dose to internal dose, or reverse dosimetry, which is from biomonitoring data from internal dose to external dose. But I can also run the benchmark dose model on an internal dose basis. This is basically the way it looks like, the input module. So you can choose your model, species, chemical, and so on. You can connect to the dashboard, but you can also user-define your chemical. The parameters can be either fixed or variable. We also have QIVV inside, and you can look at metabolism with absolute clearance, relative clearance, but also Michaelis-Menten kinetics for isoform-specific metabolism such as Km and Vmax. This is the exposure settings. So you have the dose unit, time unit, exposure metric. It could be an absolute dose, a rate, or expressed on a body weight basis. Magnitude, single or multiple dose. And then you get, once you have your chemical specific parameters, sorry, I'm allergic to this carpet, so you get your physiological parameters and chemical specific parameters. Forward dosimetry module. You have the input parameters. You simulate the model. You can also run the sensitivity analysis, model evaluation, compare your simulations, and you can get kinetic parameters and concentration in body fluids or organs. Here is a screenshot. Okay, so the types of outputs you will get with the forward dosimetry: time concentration profiles or kinetic parameters, as you can see on the left-hand side of the panel. You can also upload your own data to compare the predictions, predicted versus measured concentrations. You can also upload different simulations for different species and generate a graph to compare them. Reverse dosimetry module, you have a prior distribution, very Bayesian, and you generate a posterior distribution, and at the end you can get your exposure distribution. Here is the module, particularly useful for biomonitoring data. The toxicodynamic module, forward dosimetry results, dose response data. You can get then an internal benchmark dose limit. So it looks like. Sorry about that. So forward dosimetry results, kinetic parameters, dose response data, and you get your internal dose, benchmark dose. Dynamic energy budget model, a database connected called AdMyPet with life cycle parameters. Select the chemical, mode of action, exposure time scale, effects on individuals, and effects on population. This is what it looks like. Yeah, this dust. Okay, thank you. We also have a mixed tox module, which allows you to perform risk characterization of mixtures. Inputs are exposure metrics, hazard metrics, and you get the risk characterization performed. Finally, the automated report, which will generate your inputs and your outputs. briefly, some future perspectives. We have a project that runs in EFSA called ADME for NGRA, where we'll look at in vitro assays to study metabolism for different organs of interest to EFSA, intestine, liver, kidney, but also the microbiome. Substances to be selected for experiment under discussion at the moment. We also look at developing open access database focused on ADME properties, in silico tools on metabolism, and also improve the generic models, including integrating a microbiome compartment in those models. Future implementation of the new models, comparing performance of generic models versus chemical-specific models, that will be important to see the difference in variability and uncertainty. New models for humans, farm animals, species of ecological relevance are currently being developed. QIVV and open access distribution available for a range of metabolic pathways. Integrate those tools for QSARs and chemical-specific QIVV, of course, PBPK models. And finally, models handling chemical mixtures such as pharmacokinetic or pharmacodynamic interactions, but also we're looking at multiple stresses, for example, in bees. A lot of colleagues to acknowledge from all over the world. And with that, I'd like to thank you for your attention.
Okay, thanks for Dr. Jane Dorlon. Any questions?
Leave a microphone. So, Jean-Luc, thank you, and I mean, we really appreciate your effort. I know that's an occupational hazard there. So, Jean-Luc, I really like what I'm seeing here. So, I mean, we engage very frequently with colleagues of ours that do modeling work. My experience has always been that whenever you need their assistance, they're overwhelmed, they say, yeah, let me work on that. 6 months later, you hear, oh, do you have the half-life for this? And then you give them the half-life. And then eventually, they come up with a model. So, I mean, what I'm seeing here is just the generic framework to populate the model with our data. So, and I have not yet had the chance to see this working, but I mean, to what extent do you think that this is actually going to be accessible for, I know, someone like me? I know how to generate the basic PK data. I have no idea how to do that. So, I mean, there's parameters that are pre-populated for physiology, etc., right?
Well, so basically we have a user manual, so we basically take you through every step. And then there's a case study for every module, case study manual, so to speak, where we take you through every step for the different types of case studies you can run.
So, perhaps without wanting to abuse your availability, seeing some sort of guided example as an online training when you deploy this would be really, really good for us.
Well, that's something we are planning. So we're going to start at EFSA for the staff and the experts, but of course we can definitely do that with pleasure.
Yeah, John-Lu, thanks for the very interesting talk. I think it's very relevant for novice users who need to do modeling or understand it. Actually, 3 questions, if you don't mind. First is, do you consider transporters in the model?
Yes, we need to modify those, and that's the ADME for NGRA.
Okay.
Phase 1, Phase 2 transporters, excretion, and microbiome.
Excellent. Second question is, for the human parameters, do you also provide the inter-individual variability? I mean, I know you mentioned it at the start of your talk, but is it embedded in the model as well?
So for now, the parameters are fixed, but you can make them vary in the panel.
Okay.
It can be either fixed, and you can choose the statistical distribution you
Do you incorporate like a coefficient of variation as well, or that's user input?
So the input, the generic models these are fixed. They're like consensus value, Brown et al. 97 for humans, and so we've used those because actually a lot of people are using those, so we didn't want to reinvent the wheel.
Okay.
But yeah, this is something we are currently discussing also with the Irish colleagues to see whether we can, let's say, tier the modeling So very simple, like one compartment, HTTK, for example. The one it's actually John Wambau's model is there, the HTTK, one compartment. But we could add modules, because if we know that this compound is phase 2 only, we go straight to phase 2. Bisphenol A is phase 2. If it's phase 1, phase 2, transport, renal excretion, if we have this kind of knowledge, we could actually, refine the models in that way. That's the way we're thinking about it.
Okay, good. That kind of answers my third question, is how, to what extent you can customize it, but sounds like you can turn on and turn off various features in the model.
Hopefully, yes.
Excellent, thank you.
So, thanks for Dr. Jane. The next speaker is the last speaker of this section, is Dr. Catherine Carolou. Dr. Catherine completed his PhD in molecular biology at the University of Ottawa, and she began her current position at the Canadian Food Inspection Agency and focusing on the application of whole genome sequencing. So, Catherine, over to you.
Thank you so much. I'm a little scared to be here now. I'm really going to be changing gears here. I feel like genomics is some pretty old technology compared to some of the stuff I've seen at this meeting, but I'm going to sort of talk to you a little bit about our experience in implementing this over the past 10 years since we started. And I think it's a good example of a technology that got rolled out much faster than we expected it to. I remember giving talks, 10 years ago, and our regulators had no interest in it. They didn't think it was going to be their problem. But then it just, it went really fast. And so it's possible for this stuff to be deployed really, really quickly. But how do we make sure that we stay on top of it? And for us, it felt a little bit like building the plane while it was flying. So how do we make sure that, you don't end up in these situations? By having groups like the GCRSR, where we start to think about these things early on and try to develop approaches to deal with it. So deployment of genomics for food microbiology was actually probably the easiest use case that you could have. We only deal with really 3 bacteria. Bacterial genomes are small. And it was a pretty easy thing to start doing. We also, we test 30,000 samples a year, but we don't get many positives. Only about 1 in 100 samples are actually positive. So we were actually, by 28, so we started doing this in parallel to our traditional biochemical PFGE methods in 2014. And by 2018, we'd actually stopped doing some of the more traditional methods entirely. So we moved to this pretty quickly. And of course, this is not applied until after you actually have a colony isolate on a plate. You get, you do your food microbiology traditional method, and then you get an isolate on a plate. And, long before we've done the sequencing, we have done the recall. So we're not waiting for this data to move on with the recall action. But it does give us a whole bunch more information than we did have before. And we're able to, take a longer view of the data and get some additional use out of the food testing programs that tell us a little bit more about what kinds of things we're seeing in the food supply. And our main questions when we do get the genomic sequence data is, what is it? Have we seen it before? And is it dangerous? And so this high resolution approach has really done a great job of fingerprinting the bacteria. It helps us to link isolates to food, from food to people, or we can actually say, no, these are not related at all. We don't have to do any further investigation. But we can also start to use the data for risk assessment. There's certain listeria, for example, that we see a lot in food that we never see in people. So can we get a more refined risk analysis of, based on the bacterial genome data? And again, it's very hard to get bacteria out of food, one positive sample for every 100. So the more use we can make out of the positive samples, the more long-term benefits I think we can realize from this. And at this point, we've now shared 3,300 genomes publicly to public repositories, and it opens opportunities for global epidemiological investigation, so we can help other countries as well. And so, one of the really positive outcomes of our data sharing practices was that we submitted data that resolved an outbreak that had been going on for 3 years by the time we put that data. And there was a lot of pushback against publishing the data. People, our management didn't want to do it, so it really took us a long time to get permission to even put this in the public repository. But they've really seen this was a really good example of the kind of benefit you get from sharing that data publicly. And we've been able to use the information to have, get help with our food safety investigations. It answers questions. That really high resolution fingerprinting lets us know where the sources of contamination are coming from. Sometimes we see a bunch of different populations in a single sample, and that tells you you've got a really big problem wherever you got, rather than a point source contamination. But, there's, as I was saying, there's a lot of these downstream uses that are really valuable, and we've been using this a lot in our method development. It's really, when you go to the lab and do your method validation, it's really expensive, it's time consuming, it takes a lot of people, it takes a lot of money. So the more that we can analyze our data or model, use our big panels of 1,000 bacteria in silico before we even go to the lab, it saves us a lot of money. And recently we've been, if you can only take 15 strains through a method analysis, you want to pick the 15 most different strains that you have, and that really gives you the diversity of phenotypes that you might expect from that species. So, I'm going to move on to a little bit about what we've been doing with the GCRSR. And, when we started, we quickly realized that we're not going to get consensus about how data is going to be analyzed. Everybody was doing things in a little bit different ways. One of the nice things about this technology is the data that we're using has stayed the same for the past 10 years. So that's been really nice, and that's a really great opportunity. But the bioinformatics changes a lot. You change computers. We've moved to a cloud platform now, and so you need to be able to, and you want to make the most use of the data that you can, so you need to be agile in the way that you're using the information. So with with our GCRSR group, our bioinformatics group, we started looking at this from the perspective of, rather than trying to have rigorous validation of your rigorous parameters for your bioinformatic approaches, you use benchmark data sets where you know the answers. All of the features that you're measuring and that you're reporting on are integrated in your very comprehensive benchmark data set. And then if you change anything, then you just you can do that analysis overnight. You can do your validation automatically overnight, so you can make changes, and it's not going to impact the reliability of your results as long as you're building in those validations. So part of the work we've been doing with the GCRSR is developing these gold standard data sets that you can use for validating your pipelines. We've also developed some really terrible data sets that you can use to see if your pipelines can detect when things have gone wrong. And what we've been doing this year is actually trying to see if we can apply some of these same principles to the use of metagenomic data analysis, because that's something that's really come up a lot for a lot of people recently. We can do the single bacteria sequencing, but wouldn't it be better if we could just extract DNA from, say, a food sample, and we can see everything in there? If there's Listeria and Salmonella and VTEC, maybe we can see it all at once. And so that's the promise of it. And so this year we've been sort of trying to think about how you would, create benchmark data sets for evaluating metagenomics data analysis. So what possible issues can you run into when you are doing a metagenomic analysis? And I think one of the big ones, I think, for us has been limit of detection. With our food testing methods, we can detect one cell. I think it's going to be very hard to get a metagenomic approach that's that sensitive. So what's the limit of detection of the method? And the other thing that's an issue is, how accurate are the results? So when you see a read that says there's a salmonella in there, is there really a salmonella in there? And this is sort of where the idea of data bias comes in. So this figure shows just a sequence of E. coli, just E. coli in this sequence. But if you use the tools that we use in metagenomics, where you take each of the reads that come out of the sequence data and you map it to a database, what you start to see is that, yes, most of the reads map to salmonella, but we start to see hits to enterobacter and salmonella, and you see other species coming up as well. And that's because there's a huge data bias in genomics. There's 600,000 genomes of salmonella in the public repositories, and very little coverage of other species that you would see in food and environmental samples. So you have to keep this in mind, that when you're doing best matches to databases, these are the sort of things that you can run into. So we've been working with Carleton University on a project to see if we could get that specificity. And we focused on detection of salmonella in metagenomic samples. And what we did is we tried to, reduce the regions of the salmonella genome that we would consider to be accurate. So we had something called the species-specific regions of salmonella that we trust. So if you have a match to salmonella, then you compare it to your species-specific region. Now you're losing 90% of the reads to salmonella because they're not species-specific, so you can only use 10% of the genome at this point. But it seemed to give us better results in terms of confirmed positives when we did simulated data sets that contained salmonella reads. And this just shows you the sensitivity in a simulated metagenomic sample. And that bottom graph shows that once we have 40 salmonella reads in a library, we can actually detect that there's salmonella in there based on these salmonella-specific reads. And that the top panel shows you, if you were to use some validated gene markers instead, your sensitivity goes down quite a bit. You need 4,000 to 40,000 reads to get that detectability with a single gene. And that's just to show that, the nice thing with the metagenomics is you can use a lot more of the genome than what you're doing with a single marker. And just to follow up on that single gene problem, because another project we're working on is looking for antimicrobial resistance genes in metagenomic samples. So this is something that people want to do to evaluate the presence of AMR in different samples. And the problem is that now you've got a single gene, so you need more coverage to find that one gene in a metagenomic sample. And the other thing that happens, and you can see these different ellipses so we did simulations with different amounts of genome coverage for E. coli and Klebsiella that contained a number of AMR genes. And what you can see here, the little red circle in the middle, is that this is a measure of the repeatability of the sampling. So if you want to find exactly which gene you have, which variant of the gene you have, you need up to 10 times coverage to get a really good reproducibility of detection of that gene. And this matters for AMR because not all genes confer the same resistance. A single amino acid change can take a gene from something that has really big public health significance to something that we really don't care about because it's in very, very widespread in nature. So I wanted to show you this quickly. I'm running out of time. But we did a laboratory evaluation as well. So we also did spike-in samples. We picked feed and chicken Zika because we wanted to know what would happen if we put salmonella in a high bacterial load sample versus a low bacterial load sample. And where, you know, your salmonella would be like 1 in a million of the bacteria in the sample. So what's your detectability? And what happens is, of course, you know, if you do a direct extraction, a lot of your DNA comes from the matrix that you're extracting from. So you're already losing 90% of your reads. You can only use 10% of your salmonella genome. So depending on the background, you're really only, with salmonella, you're really, you know, in chicken Zika, we can detect 10 to the 5 salmonella. So in a sample, it's really, the sensitivity of this is really, really not very high. But the other thing that happened with this particular experiment is that the feed came up as having salmonella in even the negative samples. And we isolated Citrobacter from that sample that had the reads that mapped to salmonella. So even though we did all the work to get the specificity of the method, it still failed in the end because we don't have good coverage of environmental samples. So the conclusions from all of this is, so we've got some really great benchmark data sets that we can use and reuse to evaluate tools and bioinformatic pipelines to see how, you know, well we can, what our limit of detections are and specificity are for, in this case, just salmonella in different matrices. But, you know, it really does show that the limit of detection is, you know, pretty low with this particular method. And I'm not saying this is the best approach for doing this, but it's just sort of to provide a model of how you would evaluate the system if you were going to try to integrate it into some regulatory purpose. And so we're, within the GCRSR group, we're trying to develop these data sets that people can use to evaluate different pipelines and to get a better idea of how these methods perform in different matrices with different organisms. And so we want to develop these application-specific benchmark data sets. And I encourage you to go see TJ's poster, and she's been working on a different application with different organisms. And, you know, she's been integrating decoy species that might give you false reads in these benchmark data sets, so you can measure your performance when there are interfering organisms in the type of sample that you're analyzing. And this just sort of shows you that we want to get a rigorous approach for developing these data sets to make sure the procedures are reproducible. And just to acknowledge all the people involved in this work, I would really like to, if, you know, and maybe this is the wrong crowd here because it seems like we're all toxicologists, but we need more participation in the GCRSR bioinformatics working group. We'd like to sort of expand the network now. It's been 7 years or so that the group has been going on, and we need to revitalize this a little bit. So please contact me if you would like to become involved in this. So thank you.
Thank you, Cassie, for a very interesting insight into metagenomics and with bacterial problems and bacterial toxicology. Do we have a question? We have time for just one brief question, because we have a relatively short. Okay, over there.
Thank you very much for a very informative talk. One of the difficulty with whole genome-based analysis of foodborne pathogens is that the inability to detect whether the pathogen is alive or dead. For example, if you find a salmonella in a cooked food, what would it matter? Because you will find DNA and you will find sequence or processed food in any other way. And same with agricultural produce, for example, or in a soil or somewhere. Yeah. Can it find whether it is still viable bacteria or not, or has it been somehow attenuated or killed? Thank you.
At the end of the day, I think we would prefer to have a live organism for doing a recall action just for legal purposes. This is what we want to have. And actually we're, I didn't have too much time for this study, but at the end of the day, you'll have even if you do a bit of an enrichment and then the metagenomics, it's only one more day till you get your organism on a plate. It's not very often that we see evidence of an organism and then don't isolate it.
In terms of management, when you find an alert saying that salmonella has been found,
Yeah.
how will whole genome sequence very confidently say that you have found a viable cell and do something? For example, in an acre of potatoes, if I find one salmonella or a trace of that via DNA analysis,
Yeah.
what would I suggest the farmer to dig up all the potatoes and burn them, or what should they do?
Well,
in terms of practical utilization of the technology.
And I think this is just really, if you're going to apply this, I think it would be more for measuring, for risk assessment models or measuring whether, in different systems. I think you want to find a live organism. But if you find any, that means it's been there in your system. If you can find reliable signal. So you might want to investigate anyway.
Okay, thank you very much again, Kathy. So this concludes our first horizon scanning session of today. We have a coffee break now. Unfortunately, the coffee break is going to be relatively short because we have to ensure that we have sufficient time for the session 2. So I would like to ask you to be back here at 3:15, please. Thank you very much.